Welcome to the development of Drosophila module. In this talk, we're going to learn about gastrulation. Drosophila is a protostome that belongs to the phylum Arthropoda. All insects and arachnids come under this phylum. In the case of Drosophila, the egg and the sperm fuse to form a zygote, and that zygote then undergoes the process of cleavage to form a multinucleated embryo. The cleavage is superficial cleavage, and hence it is a type of meroblastic cleavage. Once the multinucleated embryo is formed, a process of cellularization occurs to form the blastula. During the process of gastrulation, cell movements will occur, and we end up with a segmented embryo that will hatch from the egg to form a larva. The larva then forms a pupa, which can then undergo metamorphosis to form the adult fly. Following cleavage, gastrulation occurs, and gastrulation results in the movement of cells to form the segmented body pattern of Drosophila. Gastrulation begins in the mid-blastula transition stage, and the first movements that occur involve the forming of the germ layers. Now, Drosophila is a triploblastic organism, and hence it has three germ layers: the ectoderm, the endoderm, and the mesoderm. A protein called as dorsal is expressed in the vegetal part of the embryo, and this protein signals the first morphogenetic events in gastrulation. The cells that express dorsal undergo invagination, as shown in the figure in the blue box, and this invagination results in the inward movement of cells and the formation of the mesoderm. During the first stages of gastrulation, about 1,000 cells in the ventral midline fold inward or undergo invagination to form a structure called as the ventral furrow, which will be the prospective mesoderm. So when we look at the figure that's shown, it's called panel A, and the yellow box is showing the ventral furrow. The prospective endoderm invaginates to form two pockets at the anterior and posterior end of this ventral furrow. So when we look at the figure that's now shown and focus on the yellow box, we're actually able to see the anterior end of the ventral furrow where invagination has occurred. And finally, in the last one, we can see in the yellow box, which is at the bottom, we can see the posterior end of the ventral furrow where once again invagination has occurred, and these cells are going to form the endoderm cells. Now, when invagination of prospective endoderm cells occur, the embryo forms a bent-like structure which looks more like a bean, and that causes the formation of the cephalic furrow that is shown in the yellow box in the picture to the right. The ectoderm and mesoderm cells converge and extend and migrate toward the midline to form the germ band, and the germ band forms the trunk. So when we look at the picture that's right at the bottom of the slide, the pinkish or purplish region is the germ band. Once the germ band is formed, the cells destined to form the posterior larval structures are located behind the future head, and body segments appear. Now when we look at the figure and look at the yellow box in the figure to the left, we can start seeing the body segments starting to appear. We can also see that's where all the dots are, and the labelings actually show the different body segments that are going to be forming and their locations. Now during the process of gastrulation, the germ band will retract, and this results in placing the posterior segments at the posterior tip, and we can see more well-defined segments, and thus the process of segmentation occurs. So once again, if we look at the figure, look at the black box that's there in the figure to the left, we can see the segments that will form the abdomen And the one at the top will say A8, and it goes down to A7, A6, A5, A4, and A3. Now, when we see the germ band retracts, we now have to focus our attention in the next figure. And here we can look at A8. The position of A8, if you compare it to that of the previous figure or the figure to the left of it, we can see that A8 is much more towards the posterior end in this figure than the previous one. And that is what we mean by germ band retraction. The segments are pushed back so that they are now at the posterior tip. We can see very well-formed segments once this retraction occurs, and these segments will then give rise to future structures or body parts of the fly. In this figure, we can actually see which segment corresponds to what part in the adult fly. And so, organogenesis is the process where each segment will then form a commitment to form a certain body structure. With this, we come to the end of our talk, where we learned about how cell movements result in the formation of the mesoderm and endoderm in Drosophila. And we also learned about how a germ band is formed during gastrulation and the outcome of the retraction of this germ band.